Hello, everyone. Today I'm going to talk about OpenSRH, which is our paper that was in New York's Datasets and Benchmark track in 2022. Right. So in one sentence summary of what I'm talking about, OpenSRH is the first and only publicly available SRH dataset that's out there. Right. But before I get into the dataset itself, I think it's a good idea to first introduce some background about stimulated Raman histology, some of the AI methods that we have developed with the imaging method, and then I'll go into the details about the data sets and the benchmarks. Right, so let's dive right in. So the problem that we're dealing with is in the field of neurosurgical oncology. And this is one of the most important problems in the field, and it is brain tumor diagnosis. So what happens is that a lot of these brain tumor patients will come to see a physician at the institution here, like the University of Michigan, and maybe they have suffered from a neurological deficit, such as maybe a seizure or a headache. So what happens is that they'll often get an MRI, and they all look something like this, right? So what we can see on the MRI is that we can see something's going on, right? There is a lesion here, but ultimately this doesn't give us a diagnosis, and we don't really know what this is. The only way to get a diagnosis is to obtain tissue from the tumor, right? And then from once we get the tissue, then we can put a biopsy on the tissue and then actually take a look at what the tumor is. But there are many ways to obtain these tissues, ranging from a small biopsy to a gross total resection. So what happens to these patients then? Oh, one more thing I forgot to mention is that this is very important because the diagnosis actually determines the treatment, right? So for a patient, for tumors that respond really well to radiotherapy or chemotherapy, we actually don't want to cut out all of the tumor because that ends up hurting the patient in the long run. So what happens to these patients is that they'll undergo surgery, and a specimen from the tumor will be taken. And right now, this specimen will go through a pathology workflow that is slow, labor intensive, and requires a lot of tissue processing, right? And here's what I mean. The tissue will get sent to a pathology lab, and then a histological workforce will have to process the tissue, right? And there's a lot of processing steps, including fixing, sectioning, and sometimes staining. And this requires a highly skilled workforce, and these images can also look very inconsistent sometimes. That depends on the technician's preference and their experience. And once the image is acquired, and then it will get sent to a neuropathologist to get a diagnosis. And this is a time-consuming process and can take anywhere between half an hour to up to 2 hours. And all while the surgeon is waiting in the operating room to get the diagnosis. And this is not an optical workflow. This is not an optimal workflow for us. So the question is, can we develop something better, right? We want something that's more streamlined for intraoperative pathology. An ideal workflow to evaluate this specimen should be accessible everywhere, right? Even when there's no equipment or highly trained staff available. It should be fast, right? We should be able to get the images or the diagnoses right away without having to wait for a long time. It should be standardized everywhere, right? So it should be invariant to institutional protocols, patient demographics, and the clinical workforce. And it should be accurate when compared to the existing standard of care. So it should work better than what we currently have. And I think I found something like this, and that's why we work with stimulated Raman histology. So with SRH, we can, instead of sending our specimen to a conventional lab, we can actually roll the microscope into the operating room, and we can image right there. And we can get these histology images in just under a minute. And once we have these images, we're developing AI methods to help pathologists to reach the diagnosis faster. And this whole process will take in about just a minute. Right, so we can see that there is a significant speed up from the conventional workflow to imaging with SRH. So SRH is actually currently clinically being used at the University of Michigan and many other centers across the US and also in Europe. Right, so let me go into a little more about the imaging details for how this microscope works. Right, so once we have a specimen from the patient, all we need to do is to place that onto a microscope slide. And we don't need to do any other tissue processing. We just need to put it on the microscope slide and then close the cover. Right, and then this slide can go straight into the microscope without any other processing. So once the tissue is in the microscope, we then start the image acquisition. So SRH, the technology is based on Raman scattering, right? So what Raman scattering is we have incident proton on the tissue, and it can gain or lose energy and when it's being scattered. And when it gains or loses energy, it has a shift in frequency, and we can measure this shift in frequency to characterize biochemical composition. But the problem with Raman scattering is that it's very slow to acquire the broadband spectra. So in 2008, stimulated Raman scattering was discovered, and instead of using one laser, two lasers are being used, and we can use these lasers to achieve a nonlinear amplification of a narrowband spectrum, right? So we'll choose these frequency very carefully to correspond to molecular vibrational signatures. So what we do here is we choose the two very specific Raman shifts, and they correspond to nucleic acid and the protein content in the protein and lipid content in the specimen. So once we do that, then we acquire these two channels of images of the signals that comes from the microscope. So the raw image format output is like this, and they're in their DICOM format. Once we have these images, we actually do some tissue processing, image processing for AI model training. So what happens, what we see here is the two raw channels, and we can see that the channel on the top tend to correspond to protein and lipid content that's in the tissue. And the second row here correspond to the nucleic acid, that they tend to reflect nucleic acid content. So what we can do is to subtract these two channels to highlight the nuclear contrast, and we can then concatenate that with the two channels to create a three-channel image, right? Which is something that we're used to looking at. And these images are, so these are a few examples of the patches that we generate from the whole slide images. So we will patch our whole slide image into smaller patches because these whole slide images are on the scale of gigapixels, and they're not feasible for our GPUs to process. And we use a segmentation model to determine where on the slide is tumor and where is normal. Right? So we can generate this heat map. See that here's tumors and here's the normal content. And then the tumoral patches, right? So we're confident about that these patches are tumor. Then I can go through a neural network to predict the tumor probabilities. So next up, I want to talk a little more about the AI methods that we have been developing in our lab that go with SRH images. And specifically, I want to focus on two projects. The first one is on the skull-based analysis, and it's actually featured on the cover of the Neurosurgery Journal in 2022. First, I want to talk about why we chose skull-based tumor analysis, right? Skull-based is the diagnosis of a skull-based tumor is essential to have a personalized treatment, and so it's very important for us to have these diagnoses during the surgery. And the other important bit about skull-based tumor is that it's very diverse, right? So here are some samples of normal and skull-based tissues, and we can see that these histologic images look very diverse, ranging from a very dense cellular tumors to some of the tumor that's less cellular. Right. So in this project, we use the idea that similar data should have a similar representation. So for all of these images, right, we can use a model to compute some representations for these images, which can then be used to generate the predictions for each patch. And so what we can do is we can find some similar patches, and we will pass them through a model, and we will try to make sure that they're similar on the representation space. So how do we find the patches that are similar? So we can do this in two ways. If we know the labels of our images, then we can assume that the images from the same label will have a similar representation, right? So if that's the case, then we can use the images from the same label and make sure that they have a similar representation. And if we don't know what the ground truth label is for our images, then we can actually use different augmentations, right? So for example, we can try to blur the image or maybe shift it around, right? So this way we can know that these images are still very similar to each other, and therefore they should have a similar representations. So using this technique, which is called contrastive learning, we can achieve excellent results. And as we expected, right, our supervised contrastive learning, where we know the labels during training, outperforms self-supervised methods, which is trained without labels, or the conventional cross-entropy model that's the baseline. We can also look at some confusion matrices for the models that we train, and we can see that many errors are actually in the metastases class, and that actually do correspond to our domain knowledge that metastases is a very diverse class, and it makes sense that it have this slightly worse performance. Right. We can also visualize these learned representations using t-SNE dimensionality reduction, and from here we can see that using supervised contrastive learning, we can visualize much better separation between classes compared to cross-entropy. We can see that the self-supervised learning doesn't do as well in this. That's okay, because self-supervised learning is actually trained without any supervision when we're learning the model.
Sorry, what are the points here?
They're each patches.
And like roughly how many images or patients are looking
I think in this data. So we have about 8,000 points that's visualized, but that's not all the training or the validations data set. I don't know the number of patients off the top of my head. We can touch base after. I can give you the numbers. One of the most common questions that we got from surgeon is how does that help us, right? How does the AI help us? So what we can use our AI to do is to do something called margin delineation. So what happens during surgery is that the surgeon will sample from different places on the tumor, and the surgeon will want to know where every place is sampled is tumor or normal. So our model can help the surgeon to decide that maybe different regions that they sampled is tumor, and maybe they should keep resecting, or maybe some regions that we sampled is not tumor, and maybe the resection is good. All right, so that's the first application. And the second AI that I want to talk about today is molecular diagnosis for the gliomas. So conventionally, gliomas are diagnosed with histologic features. Right? So these images will be examined by a pathologist, and they will classify the glioma grading and the subtypes based on the histologic features. And this can sometimes be subjective and could be varied between physicians, especially depends on the pathologist's experience. So in 2016, thanks to TCGA, right, there is a study that showed that actually these molecular mutations, these molecular subgroups correspond to patient overall survival and treatment response so much better than what the histologic feature enables us. Therefore, it is so that in 2021, the World Health Organization changed the way that we classify gliomas, right? From based on histologic features to only based on these mutations. But the problem here is that molecular diagnosis will require a immunohistochemistry or next generation sequencing. And these methods are very time consuming and also very, very expensive, right? So there's a need to develop an AI to help us to determine the molecular mutation for the gliomas. So the clinical relevance for having an AI method here is that it can, again, help us guide the tumor resection, right? It can help us extend access of molecular testing without requiring these expensive tests and in laboratories. And it can also help us inform clinical trial feasibility and enrollment for early post-op therapies. Right. So we call our algorithm DGlioma. So how do we train it? Right. So there are 3 steps that we need to do. We need to first learn the images. We need to learn the genomic landscape. And I'll get into what that means in just a second. And then we'll take all the components and we'll put them together to classify images using the genomic landscape that we learned. So the first step is to learn the images, right? So this figure looks very familiar, right? We'll use the same idea that we used earlier, where similar images should have similar learned representations, right? So we'll have a patch, we'll take the patches and we'll do either augmentations or we'll sample from patches that are from the same tumor class. And from here, we'll have the representations for the images. And second is to learn the genomic landscape. So what we do know about a lot of these mutations is that some of them tend to occur together, and some of them maybe don't occur together. And we actually have a lot of data about these genomic data that's actually publicly available from TCGA, CGGA, and other sources. So what we can do here is we can train a machine learning model to as a genetic encoder to basically learn these co-occurrences, and we can leverage these co-occurrences to help us predict the mutations together downstream. Right. So once we learn these 2 components, we can put everything together. So what we have is an image encoder here, and we also have our learned genomic encoder, and we'll put all the components together, and we can use a transformer architecture to help us predict the mutation of each subgroup. And once we have the classification for each subgroup, we can then combine all of them together to have an overall molecular diagnosis. We tested DGlioma in an international multicenter and prospective testing cohort, including institutions here in the United States and also in Europe. And we can see that overall, our model achieves excellent results in the 3 molecular subgroups that we are focused on, right? So these 3 subgroups are the 3 molecular mutations that the WHO have defined to help us classify the gliomas, right? We use many metrics to evaluate our machine learning model, and we can see that we achieve excellent results in over 90% for pretty much all the metrics except for ARS and ATRS. Right. And from the molecular subgroups, we can then combine them together to get the final diagnosis of the molecular classification, right? And we can see that we still achieve very good results once we combine these molecular subgroups together, and especially in patients that is under 55 years old.
Right, I'm missing the in the clinic, you take a genomic sample and then you're predicting with the histology, and you're also then predicting the genomic subtype?
No, at the testing time, we only have in the clinical setting, we only have the images.
Okay,
so
when we have that during training.
During the training, we have the labels. Yeah. a training we have the molecular labels, but in the clinic during inference time, we won't have the images. So how does that compare to the existing methods to get the molecular diagnosis, right? So one of the challenge for the current diagnosis is that the testing needs to go through immunohistochemistry or IHC. IHC tests for a very specific mutation, right? It doesn't. Yep.
Sorry, there's a question online. I just want to make sure in case it's specifically relevant. It was asked, how does it do in terms of FDR?
FDR.
Post discovery rate.
False discovery rate, I assume?
I don't know.
I'm not sure what it is.
I don't have the numbers. I'm sorry.
All right, if you can,
If you
Dominic, if you can provide a little more context in chat, or if you want to unmute yourself and ask, that would be great.
Yeah, hi. So technically, like when you if you can go back onto your last slide, again the last one, the before, yeah. So now here you are only considering the true positive rate now, like, and along with you are kind of plotting the false positive rates. In a way, you are looking into like AUC or similar kind of a stuff. So, like, do you know that if your method kind of controls false discoveries, or is the power just because like it finds like way too many options to just get considered?
I don't know. If you send me an email, I can follow up with you.
Okay, great.
With the
Thank
question.
you.
Yeah, I'm not familiar with the metric that you're talking about.
Okay.
So yeah. So let's compare how deep glioma performs compared to the current way that we're diagnosing the glioma patients. And that's through immunohistochemistry. The problem with immunohistochemistry is that it only tests for a specific mutation, and it doesn't really account for these non-canonical mutations or sometimes IDH2 mutations. So this is especially important for patients with low-grade gliomas, which is very common in patients with younger than 55 years old. And that's why it's often recommended to, in addition to immunohistochemistry, to also do sequencing, which is more expensive and takes more time. And we can see that, we can see that deep glioma outperforms the standard immunohistochemistry testing, and especially in patients that's younger than 55 years old. Deep glioma has the potential to improve the way that we do molecular testing, and in just a fraction of the time. So next up, I can show you some heat maps of how deep glioma performs. Right. So like I mentioned earlier, we kind of patch our host line image into smaller patches, and we will have a patch level prediction, and then these predictions are aggregated together to have an overall diagnosis and also this heat map that we can see. And we can, and it's very interesting to see how in these positive regions, we can see histological features that correspond to IDH mutant, such as anaplasia and other features, right? And we also see that in this region that the model predicted to be less likely as the IDH mutant. We can see that it has these microvascular proliferation, which tend to be correlated to with IDH wild type of slides. And we can also see that our model performs really well with some of these non-canonical IDH mutations, where immunohistochemistry predicted that this slide is IDH wild type, but it's actually an IDH2 mutant, right? So the immunohistochemistry does not test for that. So the deep glioma is able to predict this to be an IDH mutant as well. We have many other examples from deep glioma on our website. So if you're interested, I recommend you to check it out. All right, so I have talked about SRH and how we have developed AI methods to help to help physicians to diagnose with SRH. So next up, I want to talk about the actual OpenSRH data set. So like I mentioned earlier, OpenSRH is the first and the only publicly available SRH data set that's out there. And we selected 7 classes that are the most common, right? So that includes high-grade and low-grade gliomas, like we used in the deep glioma study. And we also included 4 other tumors that are skull-based tumors that are very diverse. And we also included normal brain IVF. And so here are some examples. And again, these patches are very diverse. We can see meningiomas and the pituitary adenomas are very cellular. And we can see some other tumor types that are less cellular. And we can see the schwannoma has their elongated cells, which is the histologic features that tend to be associated with schwannoma. And also metastases is very diverse on their own. So we divided, so we randomly selected from our training set about 60 patients per majority tumor class, and the minority classes are schwannoma and normal. So we get a lot less of them. So we selected about 20 patients for each of these classes. Right. So we randomly selected these patients from our data set, and we divided them into training and testing set. And here's kind of like the breakdown. Again, it's overall pretty balanced on the patient level, except for the 2 minority classes. Each slide is then patched into patches, and we can show a distribution. We can visualize how the number of patches and slides varies across the patients, right? And we can see that most patients tend to have 5 slides, and that is just how many slides that we're imaging during the surgery. And the differences between these 2 distributions can be caused by the specimen size, the size of the non-diagnostic regions, and also the surgeon preference. So some surgeon will sample differently than the other. Right. We can also visualize the age distribution for the patients that we released. And OpenSRH contains patients ranging from newborns to 87 years old. And we can see that different classes are all pretty well distributed among the age groups. So we can also see that some tumors tend to have different age distributions, right? For example, the low-grade gliomas tend to have a lot more in the younger patients, and that's how we expect them. And high-grade gliomas in the metastases tend to have it more in older patients.
Right. Is the age also part of the data?
Age is not released as part of data. Yeah. But if you send us an email, we can talk about collaborating and releasing the ages as well. Yeah. So next up, I wanted to show you some logistics that's associated with releasing the data, right? Not everything is just machine learning. A lot of this is actually getting the data ready. So,
Can I ask one more question?
Yeah.
Is that all data is labeled as patient level or
Yes.
patch level?
Yeah, so all the data is labeled at the patient level, and I'll get into more details in just a second. Right, so I want to talk about the website that we have created, the data use agreement, and how we're hosting the data, and how the data is organized, and also the source code that we have released with that. So I'll show a quick demo. Of the website that we have created. So this is hosted on GitHub using a Jackal template. So it basically contains the information about the paper and has some abstract on the front page. And what's important about this website is that it has some samples of the images that we can visualize, and you can zoom in to you can zoom in or zoom out on some of these images and see the cellular details for yourself. Right, so we included an example for each class that we have. So we can see these very beautiful images that you can zoom in to see all the details. Another important thing for our website is to have a a data use agreement, right? So we have a agreement to get the data. So basically anyone online can fill out this agreement, right? We basically don't provide any guarantees about the data. It's for non-commercial use. So once you fill out this agreement, you will see a screen that looks like this, right? Which will take you to the links. So we went with Google Forms, so it's pretty simple to implement, and it's also really easy to change if we need to. So as you can see, we have our data hosted on Dropbox, Google Drive, and AWS S3, right? So AWS would probably be the easiest way for someone to download this data in a very speedy fashion. But Google Drive and Dropbox has their own convenience. And we don't have the patient archive on Dropbox yet, but we will be putting a copy there really soon. So OpenSRH is sort of organized in a way that we contain the metadata file, and we also have basically the raw data for every patient in their own directories, right? So we are releasing both the raw DICOM files, so that's the raw scripts that we get from the imager, and also the patches that we have generated to train the machine learning models.
Oh, the patches data is also in DICOM.
Yes, that's right. So the patches will basically just divide the DICOM file into smaller regions, so we can actually fit that on the GPU.
It's more like ROI.
Yes, yeah. Right, so in the metadata files, we released both the patient information, we also released a train validation split. Right, so in the patient information, we released the final diagnosis, right? So high-grade low-grade glioma, so HGG here is a high-grade glioma. And we also released the results of the segmentation model that we used for prediction. Right, so these are so basically these are patches that we determined to be tumor, normal, or non-diagnostic. So we released the prediction, and these predictions are the same ones that we used to generate the benchmarks that we have with this paper. And we also have a JSON file to signify the train validation split. And this is here so that it's flexible enough so you can easily change the split if you want to, right? So this is released as part of the data set, but can be easily changed. We also have a companion source code that's released on GitHub. And our source code is implemented using PyTorch and PyTorch Lightning. So PyTorch Lightning is a wrapper over PyTorch that easily allow us to do multi-GPU training and parallelization. So going back to our demo, I can show you the repository that we have. So on GitHub, we have our basic information in our package is called OpenSRH. There are instructions on how to install these packages, and also how to run the training. And it's a pretty simple command. We just need to modify the config files and also just run the training. We provided all the components that you need to train a deep learning model, right? Including custom data sets that we need to use with the custom data sets that you need to use with the data set, right? So they're reading the JSON files and reading the images for you. So that's not something that you need to worry about. This is important because SRH isn't organized like ImageNet, like everything else, right? Because with SRH image, there is this inherent hierarchy of patients, slides, and then patches, right? So we can't just put everything in one folder, like how a lot of these data set is structured. So that's why we included our own data set.
Many deep learning models are quite costly to train on the ImageNet, those data sets. Do you have an estimated training time for like a simple model?
Yes, so the length of our benchmarks is all released as the paper, and I'll get to that in just a second. Right, and we also have the training script that, again, like I mentioned earlier, we use PyTorch Lightning, which allow us to remove a lot of the training loops and we only need to identify the inner workings of the functions. So it's very convenient, and if you're interested, I can walk through the code with you more in details later. All right, so next up, I want to talk about the benchmarks that we included with our data set. We included histologic classification, and we also benchmark contrastive representation learning on a data set. So first, let's talk about the histologic classification. So we have our image. This is basically the baseline for all of the classification tasks. We have an image and go through some kind of neural network. And then at the end of neural network, the model will predict a distribution for different type of tumors. This classification is learned with cross entropy, which maximize the similarity between the model prediction and the ground truth in basically a one-hot encoding as a histogram. We benchmarked the classification with different architectures, such as residual networks. Right, so residual network was invented in 2016. So as we see, once the convolutional network gets deeper and deeper as we add more layers, these models tend to be harder to train, right? So what ResNet did is that it added these skip connections to help the gradients to easily flow through our network, so that it's easier for us to train these very deep networks. We also included vision transformers. So vision transformer is a newer architecture that models the image as a sequence. So the images are being patched into even smaller patches. And then all of these patches are modeled as a sequence. And these sequences are learned using a transformer encoder, and it can predict the final labels with the transformer encoder. The advantages of a VIT encoder is that it has less inductive bias, and it allows us to learn these images more generally. It doesn't confine us to the convolution. But the problem with that, it requires a lot more data to overcome this low inductive bias. And it has been shown that with enough data, enough compute, VIT can outperform all the convolutional networks. So here is the benchmarks that we have. Here are the benchmarks that we have. And we can see that overall ResNet performs a lot better than VIT. And that's a potential reason is that VIT often requires a very, very large scale data sets to overcome this low inductive bias I was talking about. So efficient training of the VITs is one of the open questions in the field for both computer vision and also medical imaging. And we can again visualize the confusion matrices where again we see a lot of errors are being made in the metastases class. And again, it's because it's very diverse.
For that benchmark, did you use augmentation like you did in the contrast?
Yes. So for cross entropy, we only needed to use weak augmentation, so only flipping. Only flipping is really needed to achieve a really good result, achieve a reasonable result in the in this training, and we actually do release like details of how all the augmentations implemented in the paper. So I'll get to that in just a second. Okay. And in addition to classification benchmarks, we also perform benchmarking on these contrastive representation methods. So I'll actually like to go a little more in depth into what contrastive learning is doing, right? So there are sort of two variants of contrastive learning that we benchmarked. We have the self-supervised contrastive learning and also supervised. So like I mentioned earlier, in self-supervised contrastive learning, we don't actually have the labels for each image that we have, right? So for example, we know that these are all pictures of dogs, but since we don't know that they're dogs, right? The model will actually try to pull these two images far away on the representation space. So contrastive learning is enforced by a contrastive loss. And so we can see that in the numerator, so the numerator here is what we're trying to minimize. We can see that we use a dot product similarities for to compute how similar our positive pairs are, right? So in this case, the positive pairs are basically two augmentations of the same image, right? Since we don't know the labels. And in the denominator is what we're trying to maximize. We have a dot product similarity score of any pair, right? And we can see that this similarity score is being summed over all the samples in our mini batch, right? So that includes everything else, right? So in the catch, but also notice that there's the other dot that's here, since we don't really have this information during a self-supervised setting. And we can see that this loss is summed over all of the anchors, right? All of the images in our mini batch. And tau is a temperature hyperparameter to help us to train the model better.
Anchor is the ice image, right?
Yes, yeah. So you will be summing over every anchor in your mini batch.
What
Temperature is a hyperparameter. It basically changes the behavior of the loss function. It's a depends on different temperature. The loss function will behave differently in the way that the clusters are being formed. So you can, that's a parameter that we can tune over to basically adjust it based on the data sets that we have. Right. So in order to train self-supervised contrastive loss, we need these very strong augmentations. Right. So here is a list of all the strong augmentations that we applied. So we just apply every augmentation in sequence with a 30% chance. Right. So here are some of the resulting images. And we can see that some of them don't really look like SRH images, and that is okay because a lot of these very strong augmentations tend to do help the model to learn better for learning without a label. So we also use supervised contrastive learning, right? So in supervised contrastive learning, we actually had the labels for all of our samples, right? So we know that these are both dot images. So when we are computing our positive pairs, right, we can know that all of these images should be similar, right? So the loss function actually looks quite similar. We have identified these terms that we already talked about earlier, right? We have the dot product similarity score of a positive pair, but this time, oh, sorry, let's talk about the denominator first, right? The denominator is the same as before, right? Except in this case, we're only summing over samples in different classes. So we don't see the other dot image here. That's not being compared. And in the numerator, we're summing over all the positive samples, right? And these positive samples are acquired based on the labels of the images, right? So we know that these are images of dogs and they should be similar. Right. And like before, we are summing over all the anchors in the mini batch, and we still have our temperature parameters here as a hyperparameter. So in here is the contrastive learning metrics that we have, and we can see that overall supervised method do still perform the best because label help us learn a lot better. But we can see that with SimCLR, right? So this model is trained without any supervision without any actual labels, right? We're just looking at the images and using augmentations. And we can see that we're still achieving really good results. Another thing that we can do with these contrastive learnings is we can visualize these learned representations. And we can see that we have a much better discernible clustering in SUPCON and SimCR when compared to randomly initialized with, sorry, these are not random, these ImageNet trained initialized models. So basically these are representations learned on natural images, where we can see that there's a very big difference when we are actually trained using contrastive learning on our data with or without the need for the tumor classes. We can see some mixing on our images. So maybe things like here. So a lot of these mixing happens in images that have pretty similar histologic features, such as between high-grade and low-grade gliomas, and sometimes high-grade metastases. So as part of our benchmark, we released detailed training parameters for all the experiments that we have run. Right. So we also included the GPUs that we used and also how much time it took for us to train these models. And we can see most of these models can be trained probably within a day. So the training of this data set that tend to be quite feasible for a lot of us. All right, so in conclusion, there is still a lot of open questions that we have with our data set, right? So here are some of them, right? Domain adaptation. So a lot of existing tasks in domain adaptation it tend to be very artificial. So some of these data set is comparing the handwritten digits to the street via house numbers, right? So a lot of these tasks tend to be very artificial. And another example is taking a picture with your phone and taking a picture with a DSLR, right? So there isn't much difference here, and the tasks are all very artificial. But we can actually try to adapt SRH to the conventional H&E histology, and adapting these different methods is a very large scale benchmark that's actually based on the image properties, right, from optical imaging or from staining. There is multiple instance learning where we want to have a good strategy to learn from these weak labels that we included with our data set, right? So I mentioned earlier that we are releasing a label for every patient instead of a label for every patch, because it's unfeasible for someone to basically look at every patch, right? So we have this weak label for all of the data from the same patient, right? So how do we better leverage this label to use to train our models using multiple instance learning is another open question. In all of our benchmarks, we had a very simple aggregation method that we simply average all of our predictions, right? So a better aggregation method to get us a slide and patient level prediction is also an open question. Earlier, we looked at some of the augmented images, and in these augmentations, a lot of image doesn't really make sense, right? So how to do self-supervised learning without these augmentations or with more domain-specific augmentations is also an open question. And finally, we have seen that VIT tend to have a worse performance on our data set, and that's because these models requires a massive amount of data to train. So how to train these models efficiently, right, without a lot of data is also an open question that that is another future direction of research. So in conclusion, we can see that SRH is a rapid label-free optical imaging method that provides these very high resolution images of unprocessed biomedical specimens. The combination of SRH and modern artificial intelligence can enable us to have rapid and accurate brain tumor diagnosis. OpenSRH is the first and only publicly available data set of SRH. And we hope that OpenSRH will help promote open science, translational AI research within the field of precision oncology to optimize the surgical management of brain cancers. Right. So more details again is on our website, and I would like to thank all of our collaborators in the lab and our sponsors whose generosity make this research possible. So that's it. Any questions?
As a reminder, if you're online, you can put your questions either in the chat box or raise your electronic student hand.
Great talk, Cheng. I have two questions, maybe not even questions, but the first one goes to your simulated Raman histology. At the beginning, I remember you talking about how the reason why you pursued this project was because of variability in the technician obtaining the sample and such. You know, if you think about obtaining tissue, even for this project, is very important because this is where the entire process begins. So my question for you is, when the surgeon obtains the sample, do you have like a requirement or some kind of threshold saying this tissue sample needs to have this amount of cancerous tissue versus healthy tissue all within the same sample, or does it can't be 100%? seems like part of the tumor, doesn't matter where the surgeon takes it from, because I know, I mean, you're working with a 3D object, like if you get deeper within the tumor versus on the outskirts, you could have potentially different phenotypic histology. So what's your take on that?
Yeah, so that's really up to the surgeon.
Okay.
So sometimes a surgeon would like to sample around the tumor, right? So and that goes into the margin delineation that the AI can help solve, right? So it's really up to the surgeon on which part of the tumor that they want to have it visualized. And in general, there isn't really a lot of requirement on where the tissue come from. It can literally be anything. The imager works really well with tumor normal tissues, and we can also see like necrotic tissues and other regions, right? Some regions are very fibrous. Like we can see all of that on our images. It's just up to where does the surgeon want to see.
Up to the surgeon's discretion.
Yeah, yeah, and there are some guidelines. Maybe they're like pretty simple, like don't sample very bloody
Yeah.
things, right? So there are some guidelines, but it's really up to the surgeon.
Well, then my second simple question is, and I might have not caught it, but what is your take on your research in addressing rare tumors and being able to diagnose those?
Yeah, yeah. So a lot of the methods that we have right now is focused on the very like majority classes of tumors. But there are also strategies to identify these rare tumors. So the first thing that we can do is to basically identify the rare tumors, right? So basically during inference time, if we see a tumor that we're not trained on, right? Can we identify it, right? That's like the first step. And in addition to that, we can use a lot of these representation learning techniques or other techniques to basically help us to learn these better representations of tumors. And ideally, at the end, we can have a one shot or a few shot learning of these tumors, right? So for example, in our database, maybe we have 1 or 2 patients of a very rare tumor, right? So the goal that we're going toward with representation learning is that with only 1 or 2 patients, we can identify them separately on the representation space. So when we see them in the wild, right, we can identify these rare tumors as well. Yeah.
It's really fantastic work. For this data set, it seems like really powerful for people to go out and train their own models. And I'm curious, as people do that, if you have a way of collecting back like a leaderboard or some sort of like summary of their model performance.
Yeah, so as of now, we haven't decided on how we want to have a leaderboard of sorts. We have talked about maybe we can potentially host a competition at some point, especially with these molecular diagnosis, right? So we can release additional labels with our patients, and we can potentially host a competition to predict these additional labels, or we can release another testing set, right? To host a competition, but that's not something that we have right now, but that's something that we certainly have talked about and have planned.
So what we've seen in other contexts that really is simple and works well is at the end of your training loop example with the hyperfighting, there's a function that can just generate like a YAML just like in a format that is standardized, and then say, please send that to us, and then we can add it to the website. Like but that way you're at least getting kind of like a consistent format. And if people want to share like something that you can gather, that it's not just kind of freewheeling all over the place.
Yeah. So our code base has a pretty say thorough logging process for the training process and also evaluation. So what our code base doing that's standardized is that it saves the checkpoints, right? It also saves the state of pretty much everything. So including the optimizer or the scheduler in the data sets. And we also have a prediction pipeline that saves all of the predictions. So including like the learned representations and also the logits, but like the 7 classes. So all of that is being saved in a standardized way. Obviously, people can modify that, but we do have some kind of way that's being released with the code base to save these essential predictions in the models. So hopefully we can use something like that in the future to maybe do representation sharing or model sharing.
Any other questions? Don't see anything on, oh,
Yeah, for your deep neural model,
Mhm.
So just remind me, the ground truth comes from the IC.
So yes, so the ground truth is from IHC and also the and also the sequencing. So the ground truth that we do is the final diagnosis for each patient.
And
Yeah.
then, so what is like the concept, like, what, I guess, like, what are like the medical consequences of like your, like, classifying any of the three, like, of those gliomas?
Yeah, so different tumor will basically have a different treatment plan during surgery, right? So so in the future the goal is to integrate this AI into the microscope or into the OR, so we can inform the surgeon like right away that we think this is a specific type of glioma, and the surgeon can decide on how to proceed with the surgery.